Welcome back, guys. In this video tutorial we will going to talk about gene therapy. This will going to be a revision lecture video on gene therapy. We have discussed about gene therapy in a couple of videos earlier. So if you are a beginner and first known about gene therapy, then I will like to recommend you, please watch those videos that I have uploaded earlier. Those contains detailed information of gene therapy that will help you to understand the concept. This will going to be a small video and it will contain everything that we have discussed in details, and although it will going to be a small video, but it has the enough power to save your ass before your examination. So let's get started. So first of all, gene therapy is the novel treatment method, okay, and as its name suggests, here we are going to use the gene or mainly short oligonucleotide sequences, okay. So instead of using therapeutic drugs, now we will going to use short oligonucleotide sequences to treat disease, okay. And as here we are going to use genes, so there can be two possibilities. Upon insertion inside the body, it will going to produce such type of protein that will cure the disease, otherwise it will inhibit or it will suppress the expression of the gene, or I should say the expression of the disease gene that contribute to the disease development, okay. So with the help of those kind of short oligonucleotide sequences, we are mainly focusing to treat genetic disorders that can be hereditary or that can be acquired. Although nowadays we are also using gene therapy to treat cancers. So that is the basic introduction of gene therapy. That means here we are using short oligonucleotide as a therapeutic agent instead of conventional drug. So how we can use those kind of DNA? First of all, we will pack them in a vector, that is most of the cases retrovirus vector. Although it is not harmful retrovirus, it is modified retrovirus. Now this retrovirus will be transferred to the cell, and those cells will be transferred to the body, or in some cases this retrovirus directly transferred to the body. First type of situation where we are first transferring the gene containing the retrovirus vector inside a cell and then into the body, this kind of approach is known as ex vivo gene therapy, and where we are transferring directly the vector is in vivo gene therapy, okay. Now, so first of all, the thing you should know, that is the type of gene therapy. Depending on the type of the cell we have targeted to cure the disease, we can classify gene therapy into two parts: somatic gene therapy, germline gene therapy. Somatic gene therapy is where we are targeting somatic cells, okay. So there are various type of disadvantages in targeting somatic gene or somatic cells, okay. So what are those disadvantages? First of all, if you insert your gene of interest that will cure your disease inside a somatic cell, then the victim only get the advantage of that gene, okay, and he will recover, but his offspring, his or her offspring, will not get the opportunity to be recovered if the genetic disease is hereditary, okay. That is why most of the cases we are performing germline gene therapy, okay. That means here we are targeting sperm in case of male or eggs in case of female, okay, so that not only the victims will be cured, along with those genes will be transferred to his or her offsprings, and they will also get the benefit of that foreign gene. Okay, so that is the type of the gene therapy. So if you want to make some notes, then I would like to recommend you, please go back to those videos that we have discussed about detailedly about the gene therapy. Their notes are available. Okay, so gene therapy strategy. Now gene therapy strategy means what type of strategy we are using to cure the disease. Okay, first of all, the strategy is gene augmentation therapy. Okay, this type of strategy is useful for treating autosomal recessive disorder. Okay, so here the idea is we are inserting a particular gene's allele inside the disease cell. Okay, now autosomal recessive means, let's say, smaller is a genotype of a particular, let's say, a particular type of gene. Okay, now this, the expression of that gene creates some problem. Okay, now you are adding with the help of gap therapy a capital R. Okay, and now what Mendelian principle tells us? The Mendelian principle simply tells us that if a dominant gene available, okay, or more specifically saying, if a dominant allele is available, then the expression will be dominant, right? That means the disease will be cured, isn't it? Again I am saying, suppose smaller due to the presence of two recessive allele, there is a expression, that expression is creating problem. Okay, now you are adding a capital R allele, which is a dominant version of small r, with the help of this cat. Okay, what will happen according to the Mendelian theory? That as a dominant allele is present, the expression will be dominant type, right? And as a result, your disease will be cured. That is the concept of GAT therapy. But GAT therapy, first of all, is not possible for dominant autosomal disorders, and I think I do not have to mention it, right? Because suppose RR is present and you are replacing one R with small r with the help of GAT, it will not create any kind of change because still one capital R is present, right? And that is the reason dominant autosomal disorder cannot be cured with the help of GAT. That is one thing. Similarly, sexual, that is X-linked recessive disorder, cannot be cured. Okay, because in case of, let's say, for females, if you replace one here XR are diseased allele, okay, and small x is normal allele. If you still replace one X, there is still one XR present. That means she will still be carrier. Okay, and there is a chance, as it is sex linked, so there is a chance of that the disease will be reappear in their offsprings, right? That means here also you cannot apply GAT therapy. Okay, that is why GAT therapy is only applicable for autosomal recessive disorder. Mind it very carefully. Okay, and it involves simple addition of a functional allele. Now, another important thing is, that is we are mainly using when we are treating mainly cancer cells, that is we are simply killing those cells because we cannot cure cancer cells, right? They are already malignant. Okay, under such circumstances we will either transfer some toxin gene, which are known as suicide gene, which will facilitate the destruction of the cell, or other some cases we are transferring prodrugs. Okay, first of all, initially prodrug will not cause any kind of harm to those cancer cells. Okay, but when you will add drugs, subsequently your prodrug will be also sensitive and causes destruction of the cancer cell. Okay, So the second type of approach is already came in our exam as pro-drug therapy. This kind of situation is known as pro-drug therapy. Okay, so mind it very carefully. And another one is simple addition of toxin gene that will causes the destruction of the cell. Clear? So that is the two approach. One is gap therapy, where we are adding autosomal, where we are curing autosomal recessive disorder by simple addition of a functional allele, and second one is the targeted killing of specific cells. Okay, here we are using toxin genes or we are using prodrugs with subsequent addition of drugs. Clear? Now we will going to talk about two approaches. One is classical gene therapy, another one is non-classical gene therapy. So what is classical gene therapy? In classical gene therapy, after the therapeutic gene therapy or after the therapeutic gene delivery inside the cell, the gene will produce a protein. Okay, this protein will either fulfill the shortage of or absence of some type of proteins in the cell that will cure the disease, or in other cases it have the ability, these type of proteins have the ability to produce toxins that will kill the cell directly, or in some cases it will activate the immune system. Okay, so that the immune system can take care of those cells. Understood? That in classical gene therapy we are transferring the therapeutic genes inside the cell. The therapeutic genes will produce the protein. These proteins will either activate the immune system, and then the immune system will take care of the situation, and in some cases it will produce toxin and causes the destruction of the cell, and in some cases it will produce protein that will fulfill the shortage or absence of some type of proteins in the cell. That is why cell showing some symptoms, that is why we are getting the disease, so it will be cured. Okay, that means classical gene therapy proceeds by the expression of the protein. But in case of non-classical gene therapy focuses on the curing of the diseased gene. Okay, and that is how, or inhibition of the expression of the gene. Okay, and that is how they do their job. Okay, that means in classical gene therapy we will get the protein that will do the job for us, and in non-classical gene therapy there will be inhibition of expression of gene. For example, let's say if it is a mRNA that is giving the protein that creating some disease condition, so in non-classical gene therapy there will be, let me change the color, there will be a antisense mRNA that will inhibit the expression of that mRNA, and as a result protein synthesis will not occur. Okay, so that is the situation of non-classical gene therapy. Similar things that we have seen in case of transgenic tomatoes. Okay, now the main thing, that is the method of gene therapy. First thing is ex vivo gene therapy. What is we are doing in case of ex vivo gene therapy? Suppose let's consider I am a diseased person. Okay, I have a genetic disease that is my skin related. Okay, now what you will do, you will take cells from my skin tissues. Okay, now you will grow them in a appropriate culture media. Okay, now what you will do, with the help of a appropriate vectors, most of the cases in gene delivery, gene therapy purposes, we are using viral vectors or liposomal mediated gene delivery system. They are more standard approach. Okay, and this vector contains the gene. Okay, now you will transfect those cells, that is my skin cells, with the help of those vectors. Okay, now let's say you have constructed your gene in such a way that contains some antibiotic resistant genes. Okay, so that you can select them. So you have selected them and multiplied them in another petri plate. Okay, now you have transferred them in my body. That is the overall concept of ex vivo gene therapy. Now there is a question, why I have not used any kind of other cells? Because let's say I can also use the cell of a mouse that is readily available in laboratory. So why we are using the injection to take cells from my own body? Because remember, if we use here any kind of foreign cells, then our immune system will recognize those cells as a foreign. Okay, and immune system will start fighting against them, and as a result our entire work will be wasted. There can be a chance of that our entire work will be wasted. Okay, not only that, it can also create some negative effect in my body. Okay, that is why the most preferable way is to use autologous cells. Okay, that means the cells which are self to my body. Okay, so that my body can recognize them and recognize, yes, that is my own self. So I will not going to activate my immune system. Okay, that is why I have taken the sample from myself, and then I have inserted the gene of interest and then transferred back to from to where from where I have taken it. Okay, now there is one problem in case of ex vivo gene therapy. That is, ex vivo gene therapy is fantastic in case of those tissues from where you can easily take cells. Okay, and you can also grow them. Okay, in a very significant amount in vitro. Okay, then after transformation of those cells with the help of vectors containing gene of interest, we can also transfer back them to our body. Okay, but there are various tissues available in my body where there are also some genetic disorders. Okay, but taking cell from those tissues is not easy. Okay, not only that, if we can somehow take cell from those tissues, growing them in appropriate media will not possible. Okay, and if we somehow can also successful in that job, then after that transformation or re-implantation of those cells inside the body will not easy. Okay, under such circumstances we will going to use in vivo gene therapy. Okay, so where we are directly transferring our clone genes into the tissue of the patient. Okay, we are mostly doing this in case of tissues from where we cannot isolate the gene, or if we somehow do this, then we cannot culture them in sufficient number. Okay, and if somehow we can also succeed in that, then implantation of the cultured cell on the patient is not a joke. Okay, so under such circumstances we will use in vivo gene therapy, where we are transferring gene of interest, that is clone gene, directly into the tissues of the disease cell. The most two vectors we are using here is liposomal mediated gene delivery and viral vectors. Remember, when we are using viral vectors, we are also using along with that a special type of cultured cell that is already infected with a recombinant retrovirus. Okay, and this kind of cells are known as VPC, means vector producing cells. Okay, these vector producing cells will help to transfer the gene of interest surrounding the disease cell. Okay, now there are various type of disadvantages of in vivo gene therapy, because first thing is we are directly transferring the vectors inside our body. Okay, although it is modified vector, that viral vector not have the ability to infect us, but still, okay. On the other hand, in case of ex vivo gene therapy, we are doing the entire transformation scheme inside the host, we are doing the entire transformation scheme in vitro, and then we are transferring the transformed cell, okay, which is also an autologous cell. So ex vivo gene therapy much more safer than in vivo gene therapy. Okay, and another important thing is, in case of ex vivo gene therapy, we can select the transformed cell, okay, and then we can also amplify them, then we are transferring them inside our body. But in vivo gene therapy is not providing us such type of opportunities, and that is why the efficiency of in vivo gene therapy is much lower than ex vivo gene therapy. And the efficiency of in vivo gene therapy totally depends on the transfer of the gene and the ability of the cell to express those genes, okay. And obviously in vivo gene therapy is easy to perform, okay, but ex vivo gene therapy is quite difficult, but still it is more invasive, and our in vivo gene therapy is less invasive. So we have also constructed a difference between ex vivo and in vivo gene therapy too, right? And that will be useful for your exam. Now, up to that part, we are constantly talking about transformation of the cell. Now it is important to learn about what cell to target in case of what kind of disease, right? For suppose if your victim is a cancer patient, then you should target tumor cells, antigen presenting cell, blood progenitor cell, T cell, fibroblast, muscle cells. In case of inherited monogenetic disease, you should target lung epithelial cell, macrophage, T cell, blood progenitor cell. In case of infectious disease, you should target T cell, blood progenitor cell, APC. In case of cardiovascular disease, you should target endothelial cell, muscle cells. And in case of rheumatoid arthritis, you should target synovial lining cells. Okay, so that are the various type of target cell depending on the disease that the victim has developed. So you should keep those cells in your mind when you are performing gene therapy. And for exam purpose, I would like to recommend you to remember 3 from each category. Okay, so that is the entire concept of gene therapy that you should know for answering any question from in your exam. Okay, so first of all, gene therapy, we are using short oligonucleotide as a therapeutic agent, right? Then we are simply transferring them inside disease cell, either in vitro, and then we are transferring the transformed cell, or in vivo, okay, directly. Okay, type of gene therapy: somatic cell gene therapy or germline gene therapy. Then we have talked about gene therapy strategies: GATT, where we are curing autosomal recessive disorders by simple addition of a functional allele, or targeted killing or disease cell killing, where we are using toxin gene or prodrugs. And here we have talked about gene therapy approaches: classical gene therapy, non-classical gene therapy. Classical gene therapy involves the protein production, and then protein will help to cure the disease, and here the main goal is the inhibition of the expression of the disease gene. Then ex vivo gene therapy, in vivo gene therapy. And it is also important to know why we have to develop in vivo gene therapy: for curing those type of genetic disease that is due to defect in genes in those tissues from where isolation of cell is not possible, and if we somehow isolate them, then growing them in vitro is not possible, and then if we somehow grow them, then reimplantation of the transformed cell is not easy. That is why we are developing in vivo gene therapy, where we are transferring the clone gene directly into the disease cell containing tissue. Okay, and then here we have talked about various target cell depending on the type of the disease. Okay, so that's the video, friends. So I hope this video will help you to revive your concept before your exam. So thank you for listening to this class.